In this video, I want to introduce you to some of the anatomy of the brain, which isn't visible simply by looking at the external surface. When modern neurobiologists want to look at such kinds of internal structures, one of the most useful tools are brain imaging methods. These techniques can allow neurobiologists to see the structure and sometimes the activity in the brains of living patients. This image is an MRI, a magnetic resonance image, and it's showing us a slice of the brain through the midline. It should be immediately obvious that we're not just looking at brain tissue here, but also at the other structures that are present. So for example, you should be able to recognize the nose. Here is the tongue up here. Here's the airway. Here's the skin that's covering the surface. Underneath it lies the cranium, so the skull. And here we have some cervical vertebrae, parts of the bony bits that are protecting the spinal cord. Lying within this structure, we also have the brain and the spinal cord. Now, together, the brain and the spinal cord are called the central nervous system. And what they have in common is a developmental origin. They both differentiate from the same structure during embryogenesis. The brain and the spinal cord start as a continuous tube during embryonic development and later differentiate to become these structures. This is a different developmental origin than what we'll find for the peripheral nervous system, which will be the nerve cells and the parts of the nerve cells that lie outside the central nervous system. We see the continuity between the brainstem and the spinal cord in this image here where I've zoomed in on the foramen magnum. That's the large opening at the base of the skull that allows the nervous tissue, the medulla and the spinal cord, to remain their continuity. I think it's useful to remember the common origin of the brain and the spinal cord because they share many features. For example, the brain and the spinal cord are both surrounded by meninges. These are layers of tissue that protect the brain and the spinal cord. The meninges is one of the places in the brain where we find the cerebrospinal fluid. And cerebrospinal fluid is fluid that flows in and around the hollow spaces in the central nervous system. It plays a number of important functions, but maybe one of the most important is that it allows the brain to float a little bit like a buoy in the water, and that would prevent the weight of the brain tissue crushing the cells. Cerebrospinal fluid is found in the meninges. It's also found in some fluid-filled spaces in the brain called ventricles, and I've just highlighted two here. So this is a network of cerebrospinal fluid-filled cavities in the brain that are constantly breathing the tissue, and they can serve as important landmarks when we're looking at anatomy. Well, let's turn to the structure of the brain itself. Here's the cerebrum. Lying underneath it, hidden from what we would see if we hadn't taken a midline section, is a structure called the thalamus, the hypothalamus, the midbrain. This bump here constitutes the pons. In the back of the brain, we can see the cerebellum. At the base of the brain is the medulla, and then lying underneath it and continuous with it is the spinal cord. Okay, these different regions of the brain serve different functions. Let me talk about them a little bit more. So here is the cerebrum, this large structure, and what I've colored here initially is just the part of the cerebrum we call the cerebral cortex. It also includes major fiber tracts as part of the cerebrum, including this, the corpus callosum. The function of the cerebrum is to interpret sensory input, control complex motor behaviors, store and retrieve memory. It's the site of conscious thought, reasoning, and judgment. The thalamus is a major relay station. Almost all sensory information is processed by the thalamus, so the cerebrum wouldn't know what was going on if it didn't get that information first that had been passed through the thalamus. Hypothalamus, this is associated with drive, so if you're hungry, your hypothalamus is telling you that. If you're cold, your hypothalamus is telling you that. The hypothalamus is a primary link between the brain and the endocrine system. It's going to regulate the release of many hormones. The hypothalamus also regulates and coordinates autonomic functions, so the visceral nervous system. The midbrain is a relatively small structure, but it has a number of important functions. Most notably, it's going to integrate sensory processes such as vision and hearing. There are some important eye reflexes and auditory reflexes that involve the midbrain. The midbrain also contains several clusters of neuromodulatory neurons. For example, there's a well-known cluster called the substantia nigra, which contains dopamine-producing neurons that's present in the midbrain. The pons relays information. It relays sensory information to the thalamus. It relays information to the cerebellum. It's also involved in facial sensation, hearing, and balance, and it contains control centers for sleep, swallowing, and bladder control. The cerebellum coordinates complex motor outputs. It adjusts output from the brain to maintain balance and coordination, and it also plays a role in learning new tasks, even if they're not necessarily motor tasks. And last but not least, we have the medulla. The medulla is going to relay information between the brain to the spinal cord. It regulates breathing, vomiting, coughing, sneezing, and heart rhythms. It is essential for the housekeeping functions. It's essential for life. All right, well, before leaving this image, I want to return to what I told you earlier, that the brain and the spinal cord had developed as a continuous tube during embryogenesis. Some of the terms that get introduced in development are still used when describing the neuroanatomy in the adult, and I think it helps to know where they come from in order to be able to orient yourself. So I'm just going to close by showing you some drawings that I've made of human development. So I hope that you know that human development, embryonic development, takes about 9 months, so let's say 270 days. This is a fairly early stage then, maybe 10% of the way through development, 25 days of human development. And what we're looking at here is basically the head and the torso of the developing fetus. And at this stage, you can see the tube-like structure, but we can also see that it's starting to differentiate. So at the anterior end of the developing nervous system, we can already see a swelling that will become the forebrain. This is called the prosencephalon, or the forebrain. This is also where we find the optic vesicle, and part of the eye is actually derived from the brain. Here is the mesencephalon, what will differentiate to become the midbrain, the rhombencephalon, or the hindbrain, and the spinal cord. And at this stage, they're fairly tube-like. Move forward a few weeks to 50 days of development, and now we can see that the forebrain is split into two parts. There's the telencephalon that will give rise to the cerebrum, and also the diencephalon that will differentiate to give rise to the thalamus and the hypothalamus. Together we would still refer to these as the prosencephalon of the forebrain. Here's the mesencephalon or midbrain. Hasn't changed. But the rhombencephalon, the hindbrain, can now be distinguished as two different parts. There's what's called the metencephalon that will differentiate to give rise to the pons and the cerebellum, and the myelencephalon that will differentiate to become the medulla. And then we have the spinal cord. Moving forward even further, at about 100 days, we can now see that the telencephalon has grown so much that it's actually grown over and is hiding the thalamus and the hypothalamus. The midbrain is differentiated a little bit. You can see two characteristic bumps on the back, and we're starting to see the cerebellum differentiate. Here's the pons, the medulla, and the spinal cord. And as development continues then, the cerebrum will continue to grow, it continues to get more wrinkled, so that at the time of birth, the brain is recognizable compared to an adult brain. We have the cerebrum, the cerebellum, the pons, the medulla, and the spinal cord evident. So there's a lot of terminology here, and I think it's helpful to remember these early events in development when you're studying brain anatomy.